Hi, good morning. My name is Gabriele Messina. I'm a medical doctor. And I'm going to present you a first presentation, which is about a device which you can see at the booth E56 if you're interested. And then I'm going to show you a very short video about the product. And then finally, I'm going to present you also a research paper which has been made to prove the efficacy of the device. That's the order of my intervention. Okay, we are going to talk about StatClean. The issue about stethoscope contamination, then healthcare associated infection, about the problems, and the solution we invented. All the time you have an inspection with the stethoscope, which is the most used medical device by any health operators. You can have skin like this of the first patient, then you go to the next patient with another visit, and finally to a third patient, and so on. The point is that all the time you make this kind of visit, you get some kind of a contamination from the skin to the stethoscope membranes. So these here are 3 petri dishes. And these white dot are colony forming unit, are bacteria which are going to grow in time. And after the first inspection, this is the level of contamination you get. If you don't clean the stethoscope between the first and the second intervention, this is the amount of bacteria you get. And this is the amount of bacteria you get after the third investigation. So as you can see, the amount of bacteria is going to grow exponentially. Next patient, please. The next patient could be this patient. It's worldwide known that hand washing is the first issue to retrain, to contain hospital-acquired infection. So washing hands is the most important thing you have to do if you want to retain hospital-acquired infection. And for this reason, you find, for example, in hospital dispenser, which invite you just to clean, to disinfect your hand quite often. Has been demonstrated quite recently in 2014 by a recent paper from the Mayo Clinic that stethoscope has as much contaminated as the hands. So actually, we are talking about the same amount of contamination for which you don't do anything. Why health professionals tend to forget to clean the stethoscope, disinfect the stethoscope? Because they always are focused on treatment. They always rush between patients. And actually, it is quite simple to disinfect the stethoscope. You need some cotton and alcohol. Disinfecting the stethoscope requiring just you have to collect the alcohol, the disinfectant, and the cotton, and you have to remember to clean the stethoscope all the time. If just few numbers, if you consider the amount of expenses which are going to be wasted because of hospital-acquired infection, we are talking about something like 40 billions, which is not millions, but I'm talking about billions of dollars every year, only putting together US and Europe. I don't have any references about the rest of the world, but I prefer just to stay in the safe zone, telling you just what does the data saying every year. So every year, 40 billions of dollars going to be spent because of hospital-acquired infection globally. Considering what I've saying to you before, that hand contamination is similar to the stethoscope one, we can estimate that about 5% of those expenses could be due because of not accurate cleaning of the stethoscope, which is 2 billion per year. In the world, there are something like 30 million of health operators which use a stethoscope, and this number are going to increase. And this is our solution. Solution is StatClean, is a wearable medical device which you can put in the pocket of your coat. You simply hang the head of the stethoscope in the StatClean. It automatically starts disinfecting using UVC light, ultraviolet C light. It's 100 grams in weight, so it's very easy to carry. You simply have to attach the head of the stethoscope to the StatClean, and that's it. UVC technology has germicidal irradiation, so actually UVC can be different 3 levels of UVC of ultraviolet. A is for tanning, B is for therapeutic properties, and C has a biocidal effect, so it's killing Bacteria mold because it is destroy the DNA, so they actually are not able to replicate anymore. UVC it's a great technology with great cost ratio, and it's ecological. It eliminates any kind of microorganism without any kind of resistance. Because, for example, with some disinfectant, you can have some resistance. You don't have any resistance to UVC light. You can put the set clean on the table if you prefer. You can put it on your bag if you prefer. Anyway, very simple. Hang the head of the stethoscope. That's it. There are a light here which say that the process has started, and then when the process has finished, which lasts a few minutes, you're going to use it again safely without any problems. Little object with a lot of technology inside. We have UVC light by LED, so the latest technology at your hand. We have a microprocessor inside which regulate all the calibration to have a proper disinfection. We have memory, so you can storage how many times the device has been used, for how long, who was the person in charge to use the device. It has a rechargeable battery using a USB, micro USB, so it's very easy. You can just plug the USB that you use to recharge your telephone. It has sensor to assure that everything is working smoothly and safely for the operators. We have scientific evidence from university, and which is working. Actually, here you can see the control and the effective of the devices. Here, the amount of contamination is huge, as you can see, and only few survived, actually. We have some patents on that, some already granted, Italy, US, and others pending. You can see we are trying just to cover almost any part of the world with this technology. We are the first in the world with this kind of technology. We have the industrial know-how, technical know-how to make it, and it has an Italian design. Here in this slide, you can see the competitors. And as you can see, only the StatClean has all these good characteristics. All the other competitors use outdated technology, and actually they don't have any kind of a green approach. They use consumable. We don't use any consumable, so it is also a green technology or technology. We have scientific evidence that is working. We have published on the European Journal of Public Health, American Journal of Infectious Control, Hospital Infection, so leading journal. And the device was also awarded in several events. We have been awarded in Innovation Day in Paris. We have been the only startup selected in the CODAN, which is a German event. In 2016, in Vancouver, the device has been awarded as the best innovation device in the world for which use UVC technology. And we also had 3 seal of excellence by the European Union because of the device. The startup, of which I co-founded with other colleagues, is made by me. I'm a research professor in public health. Then there is Professor Gabriele Messina, as a bioengineer, and my brother is a lawyer. And then there is also a technician who is in charge for the lab aspects. And we are in collaboration with the Light Progress. It's a company in Siena, and Giulia Santi is the one in charge for the marketing and the distribution. You can see the device, and you can have a try at the booth. It's booth E56, so just around the corner. So you can have some more information, and you can get some brochure about that. In addition, you're going to have also a new device introduced, which is going to be introduced just in Medtech, which is StateCube. It's the desktop version of the same device. Okay, now I have just a short video that can show you how StatClean works, and then I'm going to present the research paper which I announced before. So. Okay. Okay, now you know how this works, but it's quite simple, right? Nothing too particular. Okay, now I'm going to present you this presentation, which is about the test, a clinical real test that we did in environmental hospital setting. And the aim of the research was actually to test if this device was able to be used easiness with easy by the operators, and also to be effective in a real hospital settings. That were the two objectives we wanted to achieve. The study was conducted in this clinic, which is a clinic which has about 100 beds. And from November 2016 to May 2017, we can do this crossover pre-post comparison study. We selected several words. 2 words were medicine word, permanent vegetative state word, 1 post-operating word, and 1 personal stethoscope to colleague, which were used to visit patients. So different contexts, just to have an heterogeneous place where the device could be used. Okay, you already know the device. I'm not to spend more words on that. The study procedure was meant to have some stethoscope, which are going to be used at the stat clinic, and other which we're supposed to have standard routine disinfection. So actually, there were some stethoscopes which have been treated for 5 minutes with a stat clean, where you use a contact petri dishes to see the amount of bacteria you get after the irradiation, and versus those stethoscopes which were not treated with the stat clean. So actually, we tried just to make a comparison between the two groups. And all the petri dishes were incubated for 2 days at 36 degrees. And then we make an analysis to identify which were the difference using the statistical software SPSS using the bootstrap method. In total, we got something like 272 samples. 156 samples from the not UVC treatment, and 116 samples from the UVC treatment with the stat clean. All the time, the samples were taken randomly, so people didn't know when we were in the hospital to collect the stethoscope. So actually, we sampled all the stethoscope which were attached to the stat clean, and they've been identified at the treatment, and we didn't, and we use those stethoscopes which didn't were attached to the stat clean as control. In all the words, you see in red, you see those stethoscopes which didn't have the treatment. This is the number of a colony-forming unit. And in green, you see the stethoscope which have been treated with the stat clean. As you can see, the amount of coniform unit is enormously lower when the stat clean has been used. And this is the average of all the words which have been investigated. If you shift to more technical statistical information, we got an overall decrease or reduction of about 95% with a confidence limit between 92 to 97. So actually, we reach in a real context almost a very, very important disinfection. And as you can see also in any other department, we always get a very important result. So the decrease was very, very important in any kind of a context where the stat clean has been used. So as I said, an average 95 decrease of reduction of colony-forming unit between 91 to 97%. Except for the permanent vegetative state, which was the department, the word with the highest contamination of stethoscope, our results are in line with other research which state how contaminated are the stethoscope. In particular, there is this review of S.r.l review which state that the contamination is important, and we found actually that is most important also in our example. The most critical department was the permanent vegetative state. We tried just to investigate a little bit more the reason of this problem. And it is possible that the specific bacteria flora is more resistant to chemical agent in this context, where there is this particular kind of patients. It is possible that there is insufficient effectiveness of standard disinfection or cleaning procedures in this world. Or maybe patients' modulus status create a good environmental to bacterial replication. Well, it is possible that all these answers are correct. We can definitely summarize that our experience was a positive experience because we were able to reduce the membrane contamination. And considering that the stethoscope is a non-critical object, be below 20 colony-forming units, which was the result we were able to achieve, state that the cell clean can be practically used everywhere in the hospital. We don't detect any stethoscope alteration in color or sound after the use of the stet clean. There was no loss of time by the help of the operators. They don't have to look for disinfectant or any other particular agent to disinfect it. They simply have to attach the stethoscope, as I said, to the stet clean. We don't have any consumption of medical consumables, so no wasting. We don't have any kind of bioresistance to chemical agent, which is possible all the time you use disinfectant. And we didn't detect any allergic reaction. We had an ecological approach, and it was safe for the patients and for the operators. In addition, we have to maybe highlight another important aspect, that having the stet clean on the coat, it's always a sort of symbol of hygiene. So actually, it reminds you that you have to act properly with hygiene procedures, with correct hygiene procedure all the time you do something. So in a way, you tackle the problem on the stethoscope, but at the same time, it's a gentle reminder that you have to get in habits to do correct hygiene practice. It's a visual reminder which promotes good hygiene practice. It's an involuntary education tools, so actually it's like a gentle introduction to hygiene. Another important goal is that we try to bring closer this kind of technology to the health operators and the patients, and just few limits that we had in our research. The effectiveness of the device depends on healthcare operator compliance. Of course, if I provide you the device, but you don't use it, of course, we are not able to assess that the stethoscope contamination is going to be low. So you simply have to wear it. That's it. And it is necessary to remove visual dirt if it is present on the stethoscope membrane, because UVC light doesn't just remove the dirt. It does kill the bacteria, but if it's dirty, of course, you need anyway just to remove, which is the dirty. In conclusion, the use of the automatic and wearable UVC device results in practical and easy for the health professionals, and UVC radiation on stethoscope membrane showed a marked reduction of bacterial contamination. Okay, I think I've been in time. I hope to be clear, but please, if you have any question, I'll be glad to answer to your question. And if you want to see the stet clean, you can go to booth E56 and to have a look, get some brochure, and have a talk with my colleague. Any questions? Here. That's it? Okay. Thank you for attending my presentation.